Several hundreds.
Yeah.
Shall we give them a few minutes to join?
Yeah, I think so. So are you in London now?
Yes, yeah, calling from London, and it's a very cloudy day here today.
Okay.
But it's finally gotten a little bit warmer. The weather's been terrible, and it's June, so it should be very hot, you know.
Yeah.
But
is it
that's
as
okay.
rainy as Manchester?
No, I think Manchester was rainier for sure. London's definitely a little bit hotter.
Yeah.
But yeah, sure, we're all working indoors anyway, so it doesn't really matter how good the weather is. No, it's a nice day. So it's morning time here, but for you guys it's evening, right? Just
yeah, it's about 4 o'clock.
afternoon.
Yeah.
So you'll be finishing off for the weekend?
Yeah. But we still have things to do on the weekend.
Oh yeah, okay. I know.
Yeah.
Science never ends, does it really?
You know, if it's not the pandemic, we'll have you here in Lombok.
I know. Oh well, sure. I know, that would have been amazing.
It's quite hot here, but it's quite good.
Yeah, no, I'm sure it's beautiful. I know that would be amazing. It's a real pity that we can't travel this year, and I think there's still uncertainty about conferences even next year.
Yeah, like they cancel the conference and stuff.
Yeah, or they're online, and it's not the same really because you can't meet people and, you know, discuss collaborations and things. But Zoom does make it very easy, you know.
Yeah.
Now I can be in Lombok.
Yeah, that's true. I think there's about 80 people.
Good evening for all the participants. We will Good evening, all the participants. We will start our program shortly. I noticed that there's already 69 participants and also in the YouTube. I hope that my voice I hope that my voice could be heard clearly. Good evening for all the participants in Indonesia, and I guess good evening to Dr. Catherine in UK and in London currently. Yeah. Um, Welcome to all the participants to our international lecture with the topic pharmacogenetic and rare genetic variant that will be delivered by Dr. Catherine McGurk. And this lecture will be conducting simultaneously in this room Zoom and via YouTube live YouTube. Before we move to our main session, I want to share our rules for today's lecture. Let me just share the screen. Okay, this is our rules for today's lecture. Before meeting, please check your internet connection, camera and microphone, and charge your devices. And then when you're in the meeting, please mute or turn off the microphone. And it will be nice if you can turn on your camera devices. And then move to our agenda today. We will have an opening remark from our Dean of Pharmacy, our Dean of Faculty of Medicine, Dr. Hamsu Kadrian, for opening today lecture. And the times for Dr. Hamsu Kadrian is yours.
Thank you, Bu Tika. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Thank you for giving a chance to speak, Bu Tika. Our distinguished guest, Dr. Catherine McGurk from the Imperial College of London, UK, moderator, apoteker Ankit Listia Cahyani, PhD. She is the alumni of Manchester University in UK. Also, here there is Vice Dean, Head of Pharmacy Study Program, and all participants. Before we started this guest lecture, I would like to introduce our faculty. So, our faculty consists of two major schools. The first is School of Pharmacy, and the second is School of Medicine. School of Medicine, we divide it into two sections. The first is the undergraduate, and the second is the professional stage for the medical doctor. Until today, Catherine, we have 115 lecturers, and 16 of them have a major on the pharmacy. So why is quite small? Because the pharmacy is quite young. It's only about 7 years for today. But in this year, we have opportunity to edit 4 new lecturers. So it will become 20 in the end of this year. Our vision is to be globally recognized as an institution in the field of health and medicine, with the local content in the pharmacy is ethnomedicine. This local content is related to our island, which is enriched by several flora and fauna. That we know that some of them have the pharmaceutic properties. And I think this guest lecture will be strong correlated with our vision in the pharmacy. So we know that the flora and fauna in this island may need to be explored with the biomolecular technique or science, including maybe we should explore the pharmacogenetic properties and also its genetic variant. So I think it is very interesting to be part of this guest lecture. And I would deliver many thanks to Catherine with her willingness to share with us, share his knowledge or maybe his experience on pharmacogenetic and variant of genetic of some pharmacist substance. So I think that's all from me. So I think this guest lecture will be very interesting and have some advantages for us. But I'm also look at the participant here. It is not only become from the Faculty of Medicine Mataram University, but also from several universities around Indonesia. So I think this is good opportunity to Catherine maybe to also introduce herself, and also maybe sometime maybe we could invite you to coming here to Lombok, yeah, to share with us directly here, and you
That'd be
can
amazing.
see, yeah, maybe you can see the wonderful of Lombok. I think that's all. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Hamsu, for your kind opening remark. I noticed there's a full house of participants today, 100 people in the Zoom room, and then others 30 people in the YouTube. So I hope that everyone who was staying today stay safe and healthy, knowing that the pandemic is still ongoing. Before we move to the main session, we will have a photo session first. So yes, I will screenshotting the Zoom room. There's 4 pages in here. Maybe all the participants could open the camera. I hope that all can show your face in here. Please, I will count 1, 2, 3, and then move to the second pages. 1, 2, and 3, and then moved again. 1, 2, 3, and the last page. 1, 2, 3. Yeah. Thank you all, the participants that opening your camera. All right, we will begin our main session today. And before that, I want to introduce the moderator today for the lecture. Please welcome Dr. Anggit Ristia Cahyani Sunarwidi. She is our lecturer at Department of Pharmacy from the Faculty of Medicines. Please, Dr. Anggit, the time is yours.
Thank you, Mrs. Dika, for the time, and to the committee. First of all, I'd like to welcome Dr. Catherine McGurk, and thank you so much for being with us here today to deliver a talk on pharmacogenetics and rare genetic variants. And I'd like to introduce Dr. Catherine. Dr. Catherine McGurk did her Bachelor of Arts and graduated with honors in Natural Science with a Genetics and Moderatorship in the Trinity College, Dublin, Ireland. And she did her PhD in Cardiovascular Science in the University of Manchester, UK. That's actually where I got the chance to meet Catherine. And for employment, she's an honorary lecturer in pharmacogenetics at the University of Manchester, UK. She is also a research associate bioinformatician in Imperial College London, UK. And she is also a British Heart Foundation Centre of Research Excellence Fellow in the institution of Imperial College London, United Kingdom. And Dr. Catherine has received several awards, such as Outstanding Student Representation for the Faculty of Biology, Medicine and Health at the University of Manchester for 2 years as Student Representative of 2 divisions. She also received Best Poster for Higher Scoring Abstract and Basic Science Travel Grants at the annual European Society of Cardiology Congress in Barcelona, Spain. And she also got a Best Poster presentation at the British Cardiovascular Society Spring Meeting in Manchester, United Kingdom. Dr. Catherine also has several publications in very high impact journals. One of those is Circulating Ceramides as Biomarkers of Cardiovascular Disease, Evidence from Phenotype and Genetic Studies. Without further ado, I think Dr. Catherine, the time is yours.
Thank you so much, Dr. Angeet and Dr. Kadrian and the organisers for inviting me to speak today and allowing me to share some of the topics I lecture in at Manchester and also the work we're doing at Imperial. So as Angeet mentioned, I've recently been awarded a British Heart Foundation fellowship, and that is to do with the rare genetic data at Imperial, which I'll discuss later on. But I come in my capacity today as both. So I'm going to discuss pharmacogenetics, obviously, because that'll be of interest to the Faculty of Pharmacy and Faculty of Medicine. And then just show you some of our rare genetic research in case it's of interest as well. So at Imperial, I work across two different groups, a genetics and genomics group and also a cardiac imaging group. And I'll show you examples of that data. So as Angeet mentioned, I'm from Ireland, so not actually from the UK, but we both met at the University of Manchester analysing biomarkers. So both lipid biomarkers where Angeet looked in the cellular space in the lab, while I was looking at the genetics behind them. I also had two secondments, so I worked at the University of Cape Town looking at genetic analyses of South African cohorts, and also at the Stoller Biomarker Discovery Centre, where I spent some time looking at proteomics analyses. So I moved to Imperial College London just before lockdown began and the COVID madness. But you know, with the Zoom and everything these days, it's a lot easier to do research, particularly in a bioinformatician or genetics point of view, which is a lot of software and programming. This year, I have been awarded a British Heart Foundation, so that's a charity in the UK for cardiovascular research, and 2 years funding for that as a fellow to do my own research. And I'll show you some of that towards the end. So today I thought I'll refresh you on genetics because a lot of you will have worked in genetics, particularly pharmacogenetics, but some of you this will be quite new topics or a refresher from your undergraduate or master's program. I'll also give you an overview of the UK Biobank. So this is 500,000 participants that have joined Biobank to provide their data just for research, health research. And it's a brilliant asset to cardiovascular and pharmacogenetic research. And so I wanted to highlight that because a lot of our work uses that data. I'll then discuss pharmacogenetics and the background and some gene therapy, and then look into some activities in pharmacogenetics that's going on at the minute in the UK National Health System, which is called the NHS. Then finally, I'm going to discuss a recent paper that's under review in JACC, so the Journal of American Cardiology. It's also on bioRxiv or medRxiv, about how pathogenic variants in the general population are expressed. And then also on my work in progress on the fellowship work, which is about the penetrance of pathogenic variants. But I'll describe these, so hopefully you'll be able to understand as well. So really just looking at genetics and how far it has come over the last 20 years, if we just look at even the cost of DNA sequencing per human genome, back in 2001, which is only 20 years ago, it was $100 million in the US to get one genome sequence. And you can see that this, as technology has improved, this has dropped completely. So now if you wanted to get one human genome fully sequenced, called whole genome sequencing, it costs $1,000 only. And obviously there's equivalents in different countries. This is just using the American dollar. You can see here there is a little bit of a tail off. There's new technologies constantly coming out, and there's potential that this will get even cheaper over the next few years. So in these last 20 years, we've come a huge way in our understanding of genetics. There have been three main projects that I wanted to highlight two that have been going on in the UK and one that was a global effort. So the Human Genomes Project is the first one, and that was about creating one full genome of human so that we could understand what human DNA is like in comparison to other mammals. The UK Biobank cohort, as I mentioned, is data I use a lot, so it's on 500,000 individuals, and I'll go into detail on that. And I'll just briefly mention the 100,000 Genomes Project, which is linked into the healthcare system in the UK, the NHS, for patients with rare disease and cancer. So the Human Genome Project in 2003 was a 10-year international collaborative research program with the goal of completing a full mapping and understanding of all the genes in human beings. So they found that humans have about 20,000 genes and their locations. And previous to this, scientists thought, well, humans are so much more complex than other mammals and other animals, we must have more genes. So it was estimated that we had about 60,000 genes. But actually, these 20,000 genes are about the same as a starfish. So you're probably thinking, you know, that they're obviously wrong. The difference is we have really complex gene regulation. So while we only have 20,000 genes, the other areas of the genome regulate those gene expression in a very complex manner for specific tissues, for a specific time of your life. And I'll touch on that in our basics of genetics later. So this Human Genome Project was the first time we really looked at a human genome in full, and we've learned a lot from it. So we know now that the human genome consists of 3.2 billion nucleotides, a DNA code made up of A, C, G, and T bases. Every individual across the globe, so every human being, are 99.9% identical. So we're all very similar from a DNA point of view. But obviously, because it's 3.2 billion nucleotides, there are still millions of small differences. And this is mostly random variation across the genome, but there are a few highly significant differences associated with both risk of disease and pharmacogenetics. We also learned a lot from this project, how to store all of this information. The data is quite large, how to analyze it. Different statisticians and groups across the globe created tools to analyze it, how to incorporate private sectors and address ethical, legal, and social issues. So just briefly on one of the projects that's ongoing in the UK, in 2012, the Prime Minister of the UK at the time, David Cameron, launched the 100,000 Genomes Project because his son had a rare neurological disorder. This was a project incorporating the work from 13 different genomic medicine centres across the UK. And the project now has collected, since 2018, 100,000 genomes from 85,000 people, plus 30,000 tumour genomes, because tumour DNA in cancers can be different to the germline DNA. So participants are part of the health system. They have a rare disease, and sometimes their families were recruited, or there are patients with cancer who have also been screened for known DNA markers of disease. And in this project, if a pathogenic variant is found, this can be returned to the patients to inform their clinical treatment. The UK Biobank is one of the largest publicly available data sets to date, and you can create an application globally, worldwide, depending on what you're interested in studying. So the UK Biobank is a non-profit charity funded by UK-based research councils, government bodies, charity and industry, and it's supported by the NHS. The aim was to follow the health and well-being of 500,000 volunteer participants and provide this health information to researchers from academia and industry. So these volunteers give up all of their data just to help health research. The participants at recruitment were aged 40 to 69 years old, so they were slightly older in 2000 up to 2010 from across the UK, and the aim is to continue the following of these patients and all of their records until they pass away. One of the great things about the UK Biobank is researchers are obliged to return their findings. So if you created a particular trait, or say you analyze the MRI data and you found wall thickness of the heart, for instance, you return that back to the UK Biobank so that other researchers can also benefit. So if you see publications from the UK Biobank, a lot of the time you can access that data as well. So the 500,000 participants first had an interview at the UK Biobank Centre where their population characteristics, physical measures like age, weight, obesity, BMI, etc. ECG, spirometry, eye measures, and cognitive function tests were recorded, and also biological samples were taken. And now there is access to biochemistry and metabolomics for the UK Biobank participants. They then went home and completed an online questionnaire where they described their work history, their diet and digestive health, and all of their electronic health records were synced up to the UK Biobank. So for 500,000 people, we have access to their hospital admissions, to any death registries if they were to pass away, to cancer registries if they're registered on those, and also GP data for the last 20 years. 100,000 wore an activity monitor, so if you were interested in exercise, that might be of interest. But really of more importance to our group are the last two, the imaging and the genetics data. So the aim for the UK Biobank is to have 100,000 of these 500,000 people with full imaging data, so MRI, X-ray and ultrasound. And at the minute, we've been analysing the data for 40,000 participants with heart MRI or cardiac MRI, which is a huge, really huge asset to cardiovascular genetics particularly. From a genetics point of view, all 500,000 have genotyping data. So this means we can look at common variants or common mutations that are found in these 500,000 participants, so found in at least 1 in 100 individuals, and analyse those for an influence in different traits and disease for everything measured. The aim is also to complete the cohort for exome sequencing data. So that's deep sequencing of the protein coding regions of the genome, where you can look at variants found in less than 1 in 1,000 individuals, and sometimes even less than 1 in 200,000 individuals. So very rare mutations that are found in only a handful of people that cause severe disease. Last October, we got access to 200,000 participants with exome sequencing data, and I'll show you a lot of our research that's been using that data today. The UK Biobank is also planning to do whole genome sequencing, so this would be expanding into those non-coding regions of the genome, so outside the genes, and at the minute they've only piloted that data. So this is just a good image of all of the different phenotyping data available, so the genetic data, different biological samples, MRI data, the different health and medical records, and all the different anthropometric traits. And you can actually search if you have a specific trait or disease you're interested in. You can search online on a public data showcase for the UK Biobank. So just as a funny example, if you were interested in studying the fruit and vegetables that these 500,000 individuals ate the day before their appointment, there are 60 data fields on these 500,000 people on the different fruit and veg that they ate the last day. So really that just gives you an example of really how intensively phenotyped the UK Biobank is, and the amount of data that we have to access. So briefly, I'm just going to give you a background, a bit of a refresh on genetics for anyone that hasn't looked into this field in a while. So genetics is the study of DNA, where genetics was used to thought that the rest of the DNA wasn't important. So the genetics was only the study of those 20,000 genes. But now a lot of the time you'll see genomics, which is the full analysis of the DNA. Humans have 22 pairs of autosomes, 2 sex chromosomes, so 46 chromosomes altogether. And these chromosomes are kept in the cell, where the DNA code, if you unwind the chromosomes, looks like those 4 bases that I told you. Where in the regions where genes are being transcribed, a 3 base code creates amino acid, and amino acid chain creates a protein. And in genetics, we call this genotype code for an individual, their genotype, and the expression of their genotype. So any traits that are altered because of their genetic code, or protein expression that's altered, the phenotype. So the central dogma of genetics is that DNA can replicate, but it's mostly very stable, which means it can't create protein itself. So DNA is transcribed into RNA, and RNA, which is an unstable molecule, can also replicate itself, but then is translated into amino acids, and this amino acid chain creates protein. Genes themselves are created of exons and introns. So this is just an ACTG sequence in different orders across the genome, where a promoter region and and also lots of exons and introns. And when a protein is being created, when the RNA messenger code has created this sequence, it is spliced out. So the introns here are all spliced out, and it is only the exon code that is kept, which is why a lot of the time we only look at exome sequencing. So it's just the exons. In this sequence, the 3 base pair code creates amino acids. So in this case, you have 3 amino acids that create a 3 amino acid protein. So I mentioned that for a very long period of time, we thought 95% of the human genome that wasn't coding for genes was junk DNA. And this exploration of this data was called the dark matter of the genome. But we now know that a lot of this junk DNA is actually very important in gene expression. So it's regulatory DNA. DNA can bend, so different areas of genome are important for different genes. And most of this junk DNA includes non-coding DNA, so non-coding RNA molecules and gene regulatory elements that both alter the expression of genes and so are still very important. So an example of this is, this was your gene and your promoter region. So this is the code that we used to think was important. There are plenty of areas coded downstream that can be very nearby these genes and alter the expression of the genes or be involved in the production of proteins, or they can be really far away and also can have a positive or negative role on the expression of genes. An example of this is eQTLs, so mutations that are nearby a gene, which are called cis eQTLs, that have been shown to influence the gene expression. But these eQTLs can also alter gene expression on different chromosomes or genes very far away. So obviously this makes the analysis of DNA a lot more complex. So why do we have genome variation or mutations in general? And I think this is a good question to go back to, particularly now that we're talking, with COVID, and we're talking about DNA mutations a lot at the minute. So if we just imagine strawberry plants undergo asexual reproduction. So if this is the parent plant, they create an arm and they create an exact copy of themselves in their offspring. But if a toxin or a pathogenic microorganism comes along, these can knock out, if this parent plant doesn't have resistance to this toxin or microorganism, neither will the offspring. And so therefore it could knock out the entire species. However, if you look at viruses as a good example, you might have 2 original strains of a virus, but virus can swap DNA and alter their own DNA to become slightly different strains. And so if a toxin or microorganism against the virus comes along, this might kill off the first initial parent strain of the virus, but with the shifting in genetic mutations, some of them might have resistance to these pathogenic toxins and microorganisms. And humans in this way are the same. So we have variation in our DNA for survival. And you see that even with the COVID research, how people respond differently to both the vaccines and also to having COVID. So DNA mutations come in different forms. So they're mostly inherited, and we call DNA mutations variants a lot in research, but they can be also de novo, but this is very rare. So you can get some mutations from food, drink, age, UV light, etc. But in general, they're mostly inherited and stable across your life. So if this is an example of an original DNA sequence, you can get point mutations where the T here has changed to a C. You can have inversions of the DNA, deletions where if this was coding for a protein, you'd have these 3 could be an amino acid. And then obviously here your amino acid is totally changed because of this small deletion. You can have substitutions of DNA across the genome and insertions. And again, you can have alterations to the code. So really, when we're looking at these mutations, we want to know, well, does this cause a different amino acid sequence? Is the protein function different, or is the level of the expression different? There are also larger mutations, so copy number variants that are being analyzed a lot more frequently now with the larger scale genome consortiums coming out. So copy number variants are large chunks of DNA that are inserted, repeated, or deleted, and some of them are involved in disease. You can have duplication of genes, loss of one copy or both copies of a gene. You can also have translocation. So this is an example of cancer where the two sections of chromosome 9 and chromosome 12 swap in this cancer. So it's a movement of sections of DNA from one location to another. And in some disorders, you can have extra copies of a chromosome. So for example, in Down syndrome, they have an extra copy of chromosome 21. But in general, you'll see a lot of research is on SNPs. So this is like those common variants I was talking about for the UK Biobank, where the genotyping data is used. And these are usually found in at least 1% of the population. So these are common mutations across the genome that are not always involved in disease. And the most common SNPs are called allelic variants. And these SNPs can be synonymous, they're called, where it's only the last base changes in the sequence, and that doesn't have an effect on the amino acid code. But they can also be non-synonymous, where on one of the first two bases of the amino acid code changes and you get a difference in amino acid. And this can have very different effects on the gene and the gene expression and the protein that is created from these mutations. I should note as well, though, that while a lot of studies are on non-synonymous changes to the amino acid sequence, synonymous changes can be very important too. So while they might not be important in the gene, they could also change the regulation of other genes down the stream or nearby. So when you're thinking about DNA sequencing, there's a couple of options. You could do whole genome sequencing. And whole genome sequencing is that $1,000 US for a whole genome. This gives you every base pair, all of those 3.2 billion base pairs in the human genome. But as you can imagine, $1,000 is still quite expensive. So if you wanted to analyze 50 individuals, you would need $50,000 US. And it's unlikely that with that at the minute, you wouldn't have much statistical power in only looking at 50 individuals for health research. So genotyping, which is looking at those SNPs across the genome, and particularly in Europeans, their DNA is very, very similar because we all come from less individuals that went off to Europe. And so the genotyping only needs to pick up one SNP in an area of the genome because the rest of the genome is known to be exactly the same in Europeans. And so I think that's a big reason why GWASs are usually analyzed in European DNA because they're very similar. You can do exons only or exome sequencing, which we've been working on quite a lot. And exons only obviously is about the protein coding region, if that's all you're interested in. So you're not interested in the regulation or the non-coding region of the genome. And this is called exome sequencing. And then in clinic, a lot of the time you have only panel sequencing. So they'll only have genes on a panel that are of interest to pharmacogenetics or to a specific disease. But the problem, panel sequencing would be the cheapest, but the problem with it is if our knowledge expands and new genes are thought to be involved in disease or in pharmacogenetics, you won't have the information on the panel sequencing. So once you've sequenced the DNA, you want to identify variants. So what is the predicted type of variant? Is it a missense variant? Is it a SNP? Is it non-synonymous or is it synonymous? And then you want to triage the variants. So are they already reported in disease? Are they part of normal variation, or do they alter or change the protein function and level? And the American College of Medical Genetics has created standards and guidelines for the interpretation of sequence variants. And you'll see this quite a lot in genetic research, where variants are called pathogenic, variants of unknown significance, or benign. And this is a framework for looking at the evidence for a specific variant to figure out if you think it's pathogenic or causal in disease versus if it's benign or unknown. And this takes into account how rare the variant is, and the change of that variant on the amino acid sequence, and a lot of other in silico tools. So the aim of pharmacogenetics is really, can we use genetics to improve outcomes of patients, stratify risk, and avoid harm? Can we improve the safety, efficacy, and predict risk or tailor therapeutics for the people we prescribe the drugs for. And this was a paper published, I think it was in 2012, on adverse drug reactions in the NHS, so in the UK National Health System. At the time, about 6% of hospital admissions were due to adverse drug reactions. So that's 6 in 100 individuals, which, as you can imagine, is a very large burden on the health system. This then obviously costs a lot of money to the healthcare system, so millions in medication-related admissions, millions in unused medications then if they're found to cause adverse drug reactions, and obviously very negative impact on the patient, death, severe illness, and new medications being withdrawn from clinical practice. Really importantly, it's estimated that 97% of people carry at least one variant in a gene that can influence drug response. So we need to be incorporating this information, particularly to get into more precision medicine, which I'll talk about in the next slide. Most of these adverse drug responses are due to excessive drug activity, so increased systemic exposure or increased target sensitivity. And the basics of pharmacogenetics is about looking at the effect of genetic variation on drug response, so the therapeutic effect of the drug and also the adverse effect of the drug. So there will be a group of people that at the recommended dose for a drug that a pharma company has recommended, this will work fine. But for some individuals, this will cause toxicity, and for others, this recommended dose will be ineffective at treating their symptoms. And a lot of the time, it's liver enzymes that are involved because they're highly involved in metabolism of drugs, and there are some interethnic differences, and I will discuss this a little bit in a few slides. So really, the aim of pharmacogenetics is to increase the number of people here. So having a correct dosage where the prescribed drug works well. Now, whether that is changing the drug or using a different dose for the person based on their genotype. And pharmacogenetics is an old idea. So back to Pythagoras, a Greek philosopher, he wrote in his writings about seeing a man who ate fava beans, who became unwell and passed away. And so at the time they thought it was potentially evil spirits in the fava beans, and the Romans had also written it in their texts as well. But actually this is due to an enzyme deficiency in an X-linked, so X chromosome variant that increased oxidative hydrogen in these individuals. And if they ate fava beans, because of the chemicals in fava beans, they had acute hemolytic anemia. So the aim of pharmacogenetics is to move into personalized, precision, and stratified medicine. An example of this is if you have a patient group that have the same diagnosis and the recommended prescription and dose is the same. For some individuals, this drug will be toxic, but they will have beneficial effects for their diagnosis. And for others, it will be toxic and not beneficial. Then there will be groups of individuals where the drug is not toxic, but it's also providing them no beneficial effects. And there'll be individuals that we want is this blue group. So where the drug is not toxic at the dose, but it is beneficial to their diagnosis. So the aim is to improve safety, effectiveness of treatment, and informed prescription decisions. And the reason we can do this with DNA is that, as I mentioned, DNA is inherited, so it's mostly invariable, and it's unaffected by treatment. So it's unaffected by their current prescriptions. The analysis obviously is getting cheaper, and also it's easy to obtain from blood or from a cheek swab. Pharmacogenetics and the genetic variation can affect pharmacokinetics, so drug absorption, distribution, metabolism, and excretion, and also pharmacodynamics, so modified targets, the on and off targets, and sensitivity, as well as obviously toxicity. You can have decreased first pass effects, so greater bioavailability, reduced parent drug elimination, so the drug can have a longer half-life or more few active metabolites, and an altered concentration effect relationship between good and poor metabolizers. And this metabolizer phenotype is about predicting the activity of a drug metabolizing enzyme in an individual patient based on their genetic alleles, on their genetic variants. So here's just an example of a CYP450 enzyme, CYP2D6. Now this enzyme is involved in 25% of prescribed drugs. And if we just use the example of codeine, there is 100 different allelic variants identified already, common allelic variants, where for some individuals who have different variants in this gene, in the gene for this enzyme, they'll be poor metabolizers. So if you give them codeine at the recommended dose, they will have inadequate pain relief. And this isn't a small number of individuals. You know, this is 10% of Caucasians, 1% of Asians, and 5% of African Americans. Then you'll have some individuals that have an intermediate metabolizer phenotype, so the drug works, but not very well. And then you'll have the extensive or normal metabolizer at that recommended dose. On the other side, you'll also have these ultra rapid metabolizers, so individuals that overdose at the recommended dose. And this is still 2% of Caucasians and 29% of Ethiopians based on their genetics. So if you were to treat, for example, a white British individual and give them codeine, you know, which is obviously a very common drug, 12 out of 100 of those individuals will have, and more even with the intermediates, will have altered responses to the recommended dose. And really, enzymes are affected at all different phases of drug metabolism. So at phase 1, introducing reactive or polar groups. Phase 2, different side effects like neutropenia and toxicities. Phase 3, you can have distribution of the drugs affected or side effects like cardiomyopathy and other skeletal myopathies. Drug targets can be affected. You can have unwanted side effects. And here's just some examples that are used a lot in the genetics field. So HLA variants, individuals who carry variants in these genes can have life-threatening adverse skin reactions with certain drugs, with anti-epileptics, and a hypersensitivity reaction with HIV treatment. An obvious one, I think, across the globe is the prothrombin and factor V gene variants, where women on estrogen contraceptives can have thromboembolic disorders based on their genetics. Long QT syndrome patients have sodium and potassium channel enzyme variants, and so they can get arrhythmia from a lot of regular drugs like antihistamines, antibiotics, and motility agents. So the field of pharmacogenetics, while it's ever expanding in the clinic, from a science point of view and looking at interethnic differences, this hadn't changed for a lot of years. And it was only until 2017 in Nature, a publication was published about looking at interethnic differences. So I know I said that, you know, 29% of Ethiopians can have a variant that causes them an altered codeine response. But actually, like, which is obvious to us, within the different populations, you also get lots of variability as well. So this is moving really into individual specific treatment. So it was mostly thought that rare variants with big roles in drug-related genes are found different between different populations in the globe. But we still identify rare variants within these populations with an effect on drug response. So it's personalized within populations. So I just wanted to touch on a little bit of gene therapy, and particularly some new gene therapies that are going on in the NHS. So gene therapy is treating or curing disease by altering gene expression. So this is correcting or repairing a genetic defect permanently in inherited diseases, or curing disease by targeting pathogenic processes in acquired disease like cardiovascular disease and cancer. So gene therapy, you can replace defective genes, and this is where trans genes are transferred into the body cells. So you cannot gene edit germline cells that can be inherited in offspring, so egg and sperm, or you cannot alter the DNA of an individual in the X and Y chromosomes, where they could transfer that on to the next generation. That's totally illegal across the globe. But this can be very useful in body cells where there's no cure for a disease. An example of this would be Duchenne muscular dystrophy, which is a muscle disorder due to a genetic variant where the individual has muscle weakness and passes away usually at a very young age. And if we could give them gene therapy to provide them with the DNA code that they're missing to express their stronger muscle cells, it's obviously a better cure than current treatments for muscle disorders. And one of the other additional advantages of gene therapy is the potential lifelong treatment from one injection compared to the burden on healthcare systems. There are a lot of issues with it though, so insufficient gene delivery to target cells or inadequate gene expression and adverse effects. So we need to make sure that enough of the gene is inserted to target cells in the correct position without affecting normal function. And sometimes, because viruses are used to deliver these genes, your body can have an autoimmune response against the injection of the virus in the body. And there can also be off targets. So the delivery at the minute is usually genetic virus vectors carrying the genes that are missing in the individual or need to be corrected. And there are 3 main types of viruses used for gene therapy. So retroviral viruses, there are RNA viruses that integrate into the host genome and replicate during cell division. And these can be given by direct infusion to target organs or take out the cells that are, if blood cells, for example, manipulate the genetics and reinfuse them into the patients. But the problem with these retroviruses is they can only carry small genes. So for instance, in that Duchenne muscular dystrophy example I gave you, Duchenne, the DMD gene is a very large, and so this virus won't be able to carry it. So a lot of the time they have low efficacy and efficiency, and the risk of insertional mutagenesis. So for the virus, inserting the new gene or the altered DNA elsewhere is high for RNA viruses, and this can lead to cancer. Adenoviral viruses don't integrate into the genome, so they can't replicate, but they only provide temporary effects. So they're used to treat cancer usually. And adeno-associated viruses are human DNA containing viruses that don't trigger the immune system, but again, they can only carry small genes, and there is a risk of mutagenesis. And there are now a lot of other methodologies for gene therapy that are being looked at. And this is always continually changing. So injection, liposomes, polymers, and electroporation as well. So the viral vector, you get high transfection efficiency and high, but the problems are you have problems with the immune system fighting the virus. The production cost is obviously very high, and they can only carry very small genes. While for non-viral vectors, you have much better safety profile and the production costs are lower, but they're not as good, and the treatment isn't as good for the individual. So here's an example of some gene therapies that are now being used in the UK. So in 2019, a gene therapy is now being used for age-related macular degeneration. And this is like if you had a coin in the middle of your vision and you were blind in the middle. So obviously this is really life-changing for the patients. And using this gene therapy, they inject into the back of an eye the DNA that stops the immune system from attacking retinal cells. So this is really a life-changing treatment to these patients. The NHS also in 2018 are using a personalised therapy for children with cancer. So this is for patients with advanced or progressing blood cancer with limited options or are not responding to treatment. And they take the T cells from the patient's blood, modify them in the lab to add a gene for a cell surface protein that allows their T cells then to identify the proteins on a cancer cell and then target them. So take the cells out, change them, modify them, and put them back into the bloodstream. And the T cells then, once they recognize the cancer cells, can also release cytokines and recruit the rest of the immune system for that specific cancer tumor. And so it's a one-time treatment. There's also some specific treatments for cystic fibrosis in children. And this is an example of a treatment that needs genotyping first. So this treatment only works in cases with specific mutations. And so it requires knowledge of the genotype before the prescription. And I also will talk about those, a few more examples of those in this section. So pharmacogenomics, you'll see in literature sometimes, when I said genomics analysis, usually is about the full DNA. Pharmacogenomics usually means cancer. So it's the germline or inherited DNA versus the tumor DNA. So how the tumor has acquired new mutations That's called a somatic mutation. Mutations in tumor DNA usually alter drug response, so they allow the tumor to survive or resistance against the drug or treatment that they're being given. And there are some good examples of pharmacogenetic-based therapeutics. So for example, colorectal cancer and KRAS mutations, this was the first genetic test to guide the treatment of cancer. So 30% of patients with colon cancer have KRAS mutations in this gene, KRAS. And the treatment of EGFR antibodies don't work. So giving these patients EGFR antibodies will be a total waste of money. And this is an example of a drug that's an EGFR inhibitor, and it requires genotyping. So it costs $30,000 for 8 weeks of treatment per patient, but can only work for 75% of colon cancer patients. So that is a lot of wasted money on patients where you can genotype their DNA and find out if this drug can be used to target their colon cancer. And so instead of spending this $30,000 for 8 weeks of treatment and for it to not work, they have brought in a cheaper £2,000 in the UK KRAS diagnostic test kit that for colon cancer patients you can test them first before they go on this very expensive treatment. HER2 is a really good example, so you've probably heard of this treatment as well. So the overexpression of human epidermal growth factor is usually overexpressed because of gene amplification in metastatic breast cancer, so 20%. So there is a drug that is a specific antibody for HER2 and blocks the HER2 positive cancer cells. But obviously, this has to be that overexpressed 20% of metastatic breast cancer patients. So you need to test to make sure that this antibody will work on these patients before they have access to treatment. So obviously pharmacogenetics is always, it's continually having more research, and it's still learning, we're still learning from it and trying to implement it in the clinical setting. So the Ubiquitous Pharmacogenomics Consortium are the first international implementation project in Europe, and they try to determine the impact of preemptive testing on adverse drug response frequency, severity, and the costs involved. Industry and regulatory bodies now have the opportunity to retrial abandoned compounds or reduce adverse drug reactions in marketed products, because now we know that potentially in some clinical trials, there could have been certain individuals recruited that had a mutation that altered the results of that clinical trial. And now also the pharmaceutical industry is moving away from these generic blockbuster style drugs into more expensive targeted therapies. So really in pharmacy practice, you already take into account age, weight, biochemical markers, and other comorbidities for when you're prescribing a drug, but there are still adverse drug reactions. So for example, renal function is taken into account when you're prescribing drugs eliminated by the kidneys, but can we bring in genotype to take genotype into account when drugs are being prescribed that are known to be influenced by genes? So can we give subgroup specific genotyped informed prescriptions, or an alternative therapy or an alternate dose for specific people? So why has this not been fully implemented in clinical practice yet? Well, obviously there's a lot of recent advances, so our knowledge base is incomplete. Healthcare professionals sometimes don't have an understanding of pharmacogenetics, and there is a lack of evidence-based implementation guidelines. There are a lot of ethical issues about genetic information, how you store it, how you report to your patient, and even for health insurance reasons if a patient has their genetic information. There's a lack of guidance on the requirements and consequences of testing, which deters everyone from using genetic data. And while genetics has come a very long way and we're now getting these large consortiums, the availability and expense of testing is still difficult. So it's cheaper now to sequence the genome, but you still need the equipment, the personnel, you need quality control, and some specialists, like a lot of computational programmers, to analyze the 3.2 billion nucleotides. So the role of the pharmacist in the future will be, obviously, this is going to increase the complexity of your prescribing. You'll have to now take into account genotype, age, body size, drug therapy, renal and hepatic function for some drugs. So the decisions will be affected on whether to prescribe. In some cases, the individual not only carries one variant, but also carries a couple of variants across the genome. And so obviously this makes it a lot more complex. But the key role of the pharmacist will be in interpreting genetic tests in the future. So determining the appropriate drug, changing the dose, less trial and error of drugs, and different doses for different ethnicities or groups of individuals. And so it is likely that genotyping will be brought in the future for pharmacy and prescribing. So an example of this might be predictive prescribing. So undertaking a genotype test, determining the priority of this patient. So are they high priority? Do they need an alternative drug or an alternative dose? Or are they routine priority? Can they take the standard dosing practices? Another benefit of pharmacogenetics is the reduction of polypharmacy. So polypharmacy is the use of lots of different medications by a single patient. And this is most commonly found in the elderly or those with intellectual disability, and is associated with a decreased quality of life, mobility, and cognition. So now with the use of pharmacogenetics, we could tailor drug treatments, less trial and error, and use of multiple medications, minimize adverse drug reactions, improve the treatment outcomes, and have less risk of drug interactions in these patients taking multiple drugs. And also there is a side of finance management for healthcare systems. So it's avoiding those taking lots of expensive drugs when they aren't working. So the applications are to improve drug safety and reduce these adverse drug reactions, tailor treatments to meet patients' genetics, so predisposition to response and optimal dosing, improve drug discovery, improve proof of principle for efficacy trials and clinical trials of drugs. So in Manchester, I wanted to give some examples of some activities that I've been doing that have been done in the hospital there. So Manchester Royal Infirmary have really taken pharmacogenetics in their stride. And this is an example study, the PALO study by Professor Newman and Dr. McDermott, who work in Manchester. And they have looked into neonatal sepsis. So about 90,000 children are brought to the ICU due to neonatal sepsis per year. And they're given beta-lactam antibiotics straight away for sepsis. But each year, about 180 babies are at risk of hearing loss due to being given beta-lactam antibiotics. And so they're testing now in Manchester, and I think Liverpool as well, a bedside machine for genetic testing, where they have created a specific machine that you can test for a specific variant in these babies before you give them the antibiotic. Now the antibiotic's needed straight away. So usually a genetic test would take a few weeks, but they have created this quick bedside machine that just does the genetic test in 25 minutes. And this can prevent some babies from getting that antibiotic hearing loss. They're also looking at Manchester Royal Infirmary about trialing a pharmacogenetic passport. So for instance, they are looking at, well, if they, for patients that come into the hospital, can they run a variant test on 75 specific variants that are known to really influence prescribing, and then put this information into a database, and the system can use this data to give tailored prescribing advice. And they're looking to build this into the electronic patient record system over the next few years. So for example, if you were in a hospital setting, you could put the patient's ID into a website and the drug that you plan to describe or prescribe to the patient, and it can give you some information on their variants. So this patient is an intermediate metabolizer, so it's not going to work very well for them. Antiplatelet activity will be reduced. Consider an alternative agent. So I'm just going to check the time really briefly to make sure I'm not running over. No, we've got 25 more minutes. So that is all the pharmacogenetics specific and some examples of what's going on in the clinical setting in Manchester. But I wanted to also show you some examples of the work we're doing at Imperial. And this is on rare genetic variants. So I told you about genotyping data and looking at only those mutations that are found in 1 in 100 individuals. So these are very common in the population. You'll pick them up if you analyzed 100 people, you would find at least one. But when they're very common or more common than the disease you're interested in or the pharmacogenetic effect that you're interested in, they usually have a very small effect on disease or response to drug. So what we're now moving into, due to these whole genome or whole exome sequencing technologies that give you really deep and sequencing of patients, we can now look at these rare variants. So variants that are found in less than 1 in 1,000 individuals or even rarer. So in the UK Biobank 200,000 exome data, sometimes I find variants that are only found in 1 out of 200,000 individuals. And these rare variants have a much higher impact on disease and also on pharmacogenetic alterations. So really we're moving from genetics from these common variants, and these common variants are very useful because they can point to the genes that are involved in disease, and we're moving into rare variants and mutations that are found in a few individuals but cause a very severe phenotype. So the work I've been doing at Imperial has mostly been surrounding this question: to what extent do rare variants found in the general population affect asymptomatic carriers? So if you find a very rare variant that looks to be pathogenic in the general population, but they have no symptoms yet for disease, what is the extent that these patients are going to have disease or are showing altered expression of that variant or are coming towards disease? So this is really about those American College of Medical Genetics guidelines I told you about. So the American College of Medical Genetics compiled a list of 73 genes, and this was 59 before this month, so this is just updated, for which specific mutations in these genes are known to cause diseases with defined phenotypes and are clinically actionable. But they recommend that mutations found in these genes are reported to the patient because they might be relevant and inform future clinical treatment. So for example, if you have a patient in the clinic coming for genetic testing for any reason, any disease, if you find a mutation in these 73 genes, regardless of why the genetic test was recommended, you should report it back to them. But you know, this causes a lot of confusion in the clinic. Imagine you were a patient, you know, if you're coming for maybe a cancer genetic test, and then the clinician reports to you, well, you also have a variant that's due to heart muscle problems, cardiomyopathy. What your first question is, okay, well, you know, what is my probability of disease? Am I going to get disease or am I not? And do I have an altered heart because I have this variant? What is the phenotypic expression? So this causes a lot of confusion for patients and also for clinicians. You know, what can clinicians say to these patients? Here's a list of variants that potentially might be interesting in the future. So to look into this and look into how these variants alter the expression of the gene or the risk of disease of these patients in the general population, in individuals who don't have disease, we did this in 2 different ways. So we have a paper now in bioRxiv that I mentioned, that's in submission in review with the JACC, the Journal of Cardiology in America, and also my current work on the Penetrance. So I'll discuss both of these topics. But just briefly, if you haven't come across hypertrophic cardiomyopathy, if this is the normal healthy heart, hypertrophic cardiomyopathy patients have wall thickness in their left ventricle. So their ventricle becomes very small space and they really struggle to pump enough blood around the body. And HCM is a common genetic heart disease, so it's found in 1 in 500 individuals, and it's defined by that wall thickness greater than 15 millimeters in the absence of hypertension or other valve diseases. In those clinically diagnosed with HCM, genetic testing finds a disease-causing mutation in about 50% of cases. And most of these mutations are found in 8 sarcomeric protein genes. But in the general population, many of these variants meet the criteria to be deemed pathogenic if you saw them in a HCM patient in the clinic, but they're classified as variants of unknown significance in people who don't currently have disease. But are these associated with an underlying subclinical HCM phenotype, increased wall thickness, or phenotypic variation in other ways? So I mentioned that there are 8 main genes involved in HCM where variants found contribute to this disorder. And this is the sarcomere. So they're found, these 8 genes are found in the thick and thin filaments of the sarcomere. And the sarcomere is the basic contractile unit of muscle fiber. So these slide past each other to contract in muscles. So in our paper, we looked at 200,000 individuals with the whole exome sequencing data from the UK Biobank, and also 40,000 individuals with MRI data of the heart. And the overlap of these individuals was 21,000 individuals who had both rare variant data and also MRI imaging data. So we looked at all the different variants in these 8 sarcomeric genes and looked to see if any of them have evidence for being pathogenic if you saw them in cases. And we found in the 200,000 individuals, 100 pathogenic mutations, 1,500 unknown mutations that might contribute to a phenotype, but we don't have enough evidence yet, and also 7 mutations for exclusion that cause an opposite phenotype to HCM. So dilated cardiomyopathy causes the thinning of the wall, so we excluded those from our paper. We're really lucky, particularly in the group I work with, Professor Declan O'Regan, who analyses the imaging data of the 40,000 UK Biobank participants at Imperial. And they look at this in both a 2D and 3D way. So the MRI imaging in the 2D way, they use, they segment the MRI images and create traits like wall thickness, left ventricular, the ejection fraction, so the amount of blood leaving the heart, how big the volume of the heart is, etc. And they've also, for each of the MRI scans, modeled the heart for each of the 40,000 individuals. So then we were able to say, okay, well, these people carry a potentially pathogenic variant, but these people don't have any pathogenic variants. And we can compare their composite heart structures to see, are there different areas of the heart that are involved in disease? So the results from this paper that's on bioRxiv is the prevalence of HCM in the 40,000 individuals with MRI was about 1 in 500, so very similar to previous estimates. The prevalence of pathogenic variants was 1 in 400 people carried a pathogenic variant, and 1 in 40 carried these rare, but we're not sure how pathogenic these variants are. So we looked at the 2D imaging data to see, was there an alteration in wall thickness in these individuals? So individuals in the general population carrying those 100 pathogenic variants had increased wall thickness, but interestingly, only 18% would meet the threshold that in a clinic they would be called HCM. However, they do have increased wall thickness, a subclinical phenotype. We then looked at the 3D data. So they compared this, the colors here are the comparison between those controls and the people carrying the variants. And this is the left ventricle. So it's just the structure of where the wall would be thickened, where this is the front of the structure and this is the back. These yellow lines highlight the areas of significance, and the colors are the coefficients. So positive or negative increase or decrease in wall thickness. So for those 1,500 variants, there is a small increase in wall thickness, but you can really see the dramatic increase in the significance and also the increase of the wall thickness in individuals carrying those sarcomeric pathogenic variants. And then if we look at their clinical outcomes, because using the UK Biobank data, we have their hospital and admissions and GP data, we look to see, well, are these individuals dying more? Do they have a higher risk of major adverse cardiac events? And those variant carriers do. So those pathogenic variant carriers have increased risk of death and cardiac events. And you can see this as well here, and particularly heart failure, which is known to be caused by hypertrophic cardiomyopathy. So in conclusion for this paper, pathogenic variants in HCM-associated genes are associated with a preclinical cardiomyopathy phenotype and have an increased risk of adverse cardiovascular outcomes in the general population. So the work I'm doing now at Imperial, and the work I'm working on for the fellowship with the British Heart Foundation, is about variant-specific estimations of penetrance for these rare variants found in the general population. So penetrance is the probability of developing disease given that you carry a risk mutation or an allele. So it's if you are that patient in the clinic and you're found to carry a potentially pathogenic mutation, what is your probability of developing disease over time? This is different from risk or estimations of risk, because risk looks into other factors like environmental factors and lifestyle factors. But if a variant is shown to have variable penetrance, or it's also called incomplete penetrance, it means that in the patients carrying this variant, some of them get hypertrophic cardiomyopathy and others do not. So for example, if these were 7 individuals carrying the variant, a specific variant for disease, 3 of them have clinical cardiomyopathy and 4 of them don't. So the penetrance of that variant would be 3 out of 7. So I've already told you about the clinical problem. So it's that ACMG guidelines recommending that you tell patients any variants in these genes, even though we don't know what their penetrance is, if they're going to have disease in their lifetime or not. And penetrance previously has been estimated in aggregate for genes. So looking at every variant found in a gene and putting them together and looking to see, well, genetic variants in a specific gene, what's your probability of disease given that you carry any variant. This has also been done in aggregate for disease. So penetrance for cardiomyopathy, what's your probability of cardiomyopathy? And then also there has been done for a few specific variants found in family studies, but the problem using family studies is families have very similar lifestyle, environmental factors, and also share a lot of additional genetic similarities. And so it's not really an unbiased estimate of penetrance. So I really want to know, well, what is the real penetrance of these variants if you see them in the general population? And the only way we can do this is using that UK Biobank data. So the 200,000 exons, and then also another public data set called NOMAD, which is 126,000 exons. So the hypothesis is the penetrance of rare variants in these cardiomyopathy genes can now be quantified to inform clinical interpretation for those asymptomatic carriers. And the impact is clear. You know, I showed you that 1 in 40 individuals had one of those SARC indeterminate variants or variants of unknown significance. And if we can provide an estimate of penetrance for any of those variants, it will have a huge impact on the carriers. The clinicians will be able to tell those carriers their estimated probability of disease. And for where we have data, we can look at age, sex, and ethnicity specific estimates of penetrance. And then down the line, the aim is to identify modifiers for a variant that has variable penetrance or is incomplete penetrance for a specific variant where some individuals seem to have disease and others do not. What is modifying? What is putting them over the threshold for disease? Is it additional genetic factors that they have across their genome that is putting them over, that gives them disease while it doesn't give other people disease that carry this specific variant? Or is it environmental factors for cardiovascular disease, lifestyle factors, etc.? And then this can be undertaken for any disease. So we're identifying best statistical approaches for the field and also a web-based tool for people to both access our data and complete calculations online. So I wouldn't be able to do this project without my current position in the cardiovascular genetics group at Imperial, where we have access to 10,000 HCM patients and their genetic data and 2,500 dilated cardiomyopathy patients, another cardiomyopathy that's quite common. We have genetic data from the UK, US, Singapore, and Egypt. And so we use this data then to estimate the penetrance of variants found in these patients. So from a statistical point of view, I told you that the penetrance is the probability of developing disease given that you carry these variants. And so we can use Bayes' theorem where D is disease and A is a variant or an allele. So penetrance here is the probability of disease given that you carry an allele. And in this Bayes equation, you can work it out by looking at the probability of disease. So here we're using the baseline risk of disease in general population, which is 1 in 500 for hypertrophic cardiomyopathy. This is multiplied by the probability of the allele given disease. So this is the allele frequency in those 10,000 HCM patients divided by the probability of the allele in the general population. And so that's using the UK Biobank and that NOMAD data to estimate the probability of the allele. And so these are just preliminary HCM results that are unpublished currently. So this is just looking at that initial hypertrophic cardiomyopathy disease. In our analysis of 10,000 cases, we found 1,300 rare protein altering variants in 38% of those cases. Most of the variants we found are private to a single individual, but a very important portion are recurrent. So 72 variants were seen more than 10 times. So these would be important for 40% of our genotype positive HCM cases. So if we can just estimate even just for those 72 variants, we can provide a huge amount of information for genetic positive HCM cases. And so this is just an example plot of the preliminary data. So across the x-axis, it's the count of how many times we saw the variant in our cohort, and the penetrance is on the left, where this is 10% penetrance and above. ClinVar is a database where clinicians report if they see a specific variant in a case. And so some of them on ClinVar are known to have really strong evidence to be pathogenic, and others have less evidence. So if we look at, say, this specific variant, this variant has slightly less evidence, but now we're finding that individuals who carry this variant have greater than 10% probability of having hypertrophic cardiomyopathy. And there are also these variants. So this red one here, it is definitely involved in disease, but you can see that if you carry this variant, your probability of disease is less than 10%. So this can really help to inform clinical practice and also our understanding of specific rare variants. So the obvious question for future work is, well, why do these penetrant variants vary, and why does penetrance vary in these rare variants? So we can look into the protein domains, the position, the consequence that these variants have on the protein. For instance, if there was like specific areas where the variants were more penetrant, or specific functional groups that the penetrant variants were found in, we might be able to look at underlying biology. And also we can team up with our imaging collaboration on those 2D and 3D models to see how specific variants are found in the general population and what they look like in the carriers. So thank you so much for all listening and joining this webinar today. So I've covered obviously a lot of topics in a pretty short period of time, but looking forward to your discussions and your thoughts and any questions on this work. And just briefly, I'd like to acknowledge the British Heart Foundation, a cardiovascular charity in the UK, for funding me, and particularly for my fellowship for the next 2 years. And also the group leads of the 2 groups I'm part of at Imperial. So Dr. James Ware on the cardiovascular genetics group, and Declan O'Regan and all the team at the computational cardiac imaging group. And also to our external collaborators who have been part of this data. So thank you very much.
Thank you so much, Catherine. That was a very interesting talk. And it's also something that we in Indonesia is looking forward to be able to do, like pharmacogenetics, to have like more precise medication for patients. And we have a lot of questions from the audience.
Great.
And the first one will be from, I would like to ask the committee, would you show the, are you going to show the questions or? Okay, I think I'll just read them out. So the first one is from Arif Setiawansha. He was actually one of our students here, and he graduated and he's now a master's student in Bandung. And his question is, as we know that cells, RNA and DNA-based therapy can either repair and damage cells or induce the cell death or cell apoptosis in some disease. However, some patients that use these therapies might need more than one treatment. And even some patients would not be cured, or the treatment would lead to death of the patient. His question is, why did this phenomenon happen? And is there any correlation between this phenomenon and rare genetic variants?
Great question. So, yeah, really, it's a really interesting one. So if we pick a specific case study, maybe it'd be the easiest way of looking into this. So, for example, there is an immunodeficiency disorder called SCID, SCID, where it was termed in the media bubble boy. I think got a lot of media access when this all came out. I think it was about 10 years ago, maybe longer. Where the patient was born and didn't have the DNA to create white blood cells. So they had to live in a bubble of obviously very clean air, and all their food had to be very clean, because even the most basic flus and colds could be really detrimental to this patient, who basically didn't have a working immune system. To treat this, they tried to give patients the gene they were missing to create functional white blood cells. And they used gene therapy by injecting a virus into the system and providing that genetic DNA that they were missing. Now, this worked in some patients, but in others, there was massive adverse reactions, as this student has noted, and can lead to death of the patient. So I did mention that some of these virus vectors can have a huge immune response and lead to death. So there is a risk. Now, gene therapy has come a very long way over the last few years, but it's still obviously, it's not common practice yet. And even in the examples I showed, they're quite rare examples for very detrimental illnesses, like the blindness in age-related macular degeneration. For that, for us to be able to predict, though, who is going to have a response, it's a bit trickier. And at the minute, it depends on what disease you're looking at and what treatment you're looking at, and how good the genetic analyses have been. But these large-scale genetic consortiums will really help with that. But obviously you're right. So do we know at the minute what the specific rare genetic variants are to patients overreacting to certain treatment? Not for everything, for sure. There's a couple of things, if you were interested in doing this, there's a couple of things you'd have to think about. You'd have to collect enough individuals that you'd have statistical power to look into this. And you'd also have to have funding for whole genome or whole exome sequencing to analyze their data. But this is a great question, and it's similar for even the COVID vaccine responses and the response to the disease, how some individuals have no symptoms and others have very severe symptoms. And I think as more and more individuals have genetic sequencing done, we'll learn more and more about both common and rare genetic variants, and be able to pick out those individuals at particular risk.
Thank you, Catherine. I'd like to ask Arif, are you here with us? Okay, I think I'll move on to the next question. Then it's from Rifa Hartini. She's from Al Azhar Islamic University. It's another university in Mataram. And so in clinical pharmacogenetics, as already explained, it requires patient genetic data to determine therapeutic steps and drug effectiveness. This is also done in the Human Genome Project. We know that the DNA can reveal a lot of personal information, and in medical practice in Indonesia, the patient's autonomy is still a priority in terms of treatments. This will certainly raise issues. First of the information can be used by irresponsible people. And secondly, the patient's autonomy rights isn't important, or in this case, the patient doesn't have the autonomy when it comes to making decisions for their own treatment. What would you say about this? Thank you.
Really good point. And I didn't really delve into all the ethical issues to do with DNA testing, but you can imagine that there's huge ethical issues, and even RIFA has raised them here, both to do with how you handle the data and how you report it to the patient, but also to do with health insurance and what individuals want to do with that information. So I do, in general, if we just look at health insurance initially and looking at that, there are some, if you've had a genetic test, there, as we go on, sometimes health insurers will ask, have you been tested for Huntington's disease, particularly, which is a disease that's not curable at the minute. And if so, they might alter your health insurance and not provide it if you have specific mutations that lead to Huntington's disease. In general, unless you are in the clinic and you get specific genetic counselling to ensure that not only are you understand all the implications of having a genetic test, but you also can handle the results of a genetic test. In the clinic, it's usually genetic counselling and then providing the individual with the result. An area where this is a bit blurred would be like the 23andMe or all those direct-to-consumer testing kits, we call them. So in a lecture I give in pharmacogenetics, I highlight a lot of these, where you can spend money, they'll take your DNA and they'll give you some information. So 23andMe is one of the most important, it's one of the most looked at ones because the FDA have regulated it. So you can pay, I think it's like $100, send your DNA, and 23andMe will provide you with some information about your carrier status for diseases like cystic fibrosis. But the genetic counselling doesn't come before these tests. So do these people, who buy each other a 23andMe kit for Christmas or, for a birthday, really understand the implications of having that genetic information? I'm not so sure. The other thing is with 23andMe is if you take part in research, they do release a lot of great research, but then they kind of have a bit more control over your DNA in the future. There is also, in the cohorts I've spoken about today, these individuals have volunteered or have wanted to be part of, so they have all consented to be part of these big biobanks and research consortiums. And so, I don't think in the clinic even they would do genetic testing unless the patient wanted to find out the result. But there is this pipeline of you have to have genetic counselling to make sure they understand everything, do the test, and then also understand the results, because this can also have implications on family and other implications to your health and lifestyle. In general, though, the variants that people find out about are quite useful. Like if you find out you were at risk of certain cardiovascular disease, you could alter your lifestyle, exercise more, eat more healthily, and be more aware of your decisions if you're more at risk of disease. And so there are lots of patients and people who send away for these kits who do want to know this information. For anonymity of these patients in the UK Biobank and others, the researchers have no access to the patient information. So, it's totally illegal, and we abide by all the information and all the contracts that UK Biobank have, so we don't ever try and look in to see if we can identify patients. They're all anonymous, and some of the specific information about them is withheld as well, so it's not for general analysis. So I hope that answers your question, but you can even read, there's plenty of literature on all the different ethical issues with DNA testing, and as we advance in genetic analysis in the future.
Thank you, Heather. I was actually thinking of doing the 23 testing, but I didn't end up doing it. But it was very advertised well about giving you full genetic information.
When they first came out, the FDA stopped them and did a full check to make sure all their processes were correct. So they are one of the best ones to go for. But I have seen some crazy genetic tests where you can send away your DNA for like £100 and they'll tell you how good a soccer player you will be, or what your favorite wine will be, or they'll do like a dating thing where they'll set you up with someone who has complementary DNA, which doesn't even make sense.
Yeah.
So, but what I think those companies are doing is they get your DNA and then they can sell it on to pharma companies. Usually that's what happens, because how can you, like, what are the genetic mutations that say what kind of wine you're going to enjoy? Like, it's just madness. But 23andMe is definitely, I think, one of the best, and they do publish a lot of really useful research to the field with the data that they get, so it's not all for pharma.
Okay. So we still have a lot of questions. So the next one is from, I'll try to check. From Jakarta. So it's from Naswa. If you have had cancer, will your chances of getting other types of cancer increase? Thank you.
I think this is more of a question for clinicians. There's no way to know. So this is actually a good point to discuss environmental factors. So you might have heard of the term heritability, which can be quite confusing for individuals. Heritability is just about in a certain trait. So for instance, cancer incidence, how some people have severe cancer and some don't. And it looks at the pattern within families to see how inherited these certain cancers are. That is on a scale of about 0 to 100%. So for instance, height is really inherited. Height is about 80% inherited, which means that your DNA is going to have a big influence on your height. It's how much of the variation in the trait is due to genetic factors across humans. The other 20% though, however, is about environmental factors. So this could be, you might have the DNA for height, but if you're not provided enough nutrition at birth, you might not be a tall person. You might have the DNA for height, but you might also have scoliosis, which means that actually the measurement of height is a lot lower. There's also, you know, in that environmental 20%, it could be measurement error. Maybe people measured 1,000 people for their height, and some people had shoes on, some people didn't. So there's a certain amount of each trait that's due to genetics, but there's always an amount of environmental factors. So if your family member had cancer, it doesn't necessarily mean you're going to have cancer either. But I think that your chance of getting cancer is very specific to the individual, and you're better off speaking with clinicians if you're worried about your type of cancer.
Thank you. Moving on to the next question. It's from Hasriatun Padni. Oh, this is actually my research assistant.
Great.
So she was asking, let me introduce yourself. My name is Hasriatun Padni from the Biology Department, University of Mataram. My question is how the SNPs gene can cause genetic variation in the population.
Okay, so how SNPs cause genetic variation in the population? So SNPs are common mutations. So some of them don't have a role in disease and some of them do. For SNPs, because they're very common, we only look at SNPs that are found in greater than 1 in 100 individuals. So let's just imagine I have a specific SNP that is that alters the gene expression for I don't know, hair color. This specific SNP, because it's so common, it's not going to have a massive effect on hair color, but it might slightly increase or decrease the color of your hair. The role of the SNP in 1 in 100 individuals might just, so this is a good question. So the 1 in 100 means that in our reference human genomes, the 99 people have the same variant, but this one person has a slightly different mutation. So if that one person is seen with a slightly different mutation, they'll have a slightly different expression of the gene in SNPs that are known to be involved in gene. There are some SNPs across genome that we don't know what their role is yet. But that's why we do genome-wide association studies, which are looking at every SNP across the genome. And then you can look at the trait in an individual. So say you had 1,000 people and you were interested in height, you could measure everyone's height. So you'd have some people who are really tall, some people who aren't, and some people who are really short. And then you can compare their genetics, and any variant that associates with this high, medium, low will be significant. For example, some people in a SNP, you have 2 copies of every chromosome. So at a specific SNP, you'll have some people who have the reference. So it looks, majority of individuals have this reference. So it could be like an A base, and then the SNP could be going to a G. The people who have the A might all be very tall, and then the people who have 1G and 1A might be that medium height, and then the people who have the 2Gs might be really short. And so when you do a statistical analysis, it's like a t-test across the SNPs of the whole of the genome, where that pattern of A, AA, AG, and GG associates with how tall or how short an individual is. That's what in general we use SNPs for at the minute. And then when we find a SNP nearby a gene, we can investigate that gene for severe disease or rare variants that are causing big change in height. I hope that answers your question on SNPs.
It does affect, does it give? Okay, before I move on to the next question, I'd just like to welcome any of the audience if you want to ask directly to Dr. Catherine. Okay, while waiting, I'll read on the next questions. It's from YouTube. So because we only have 100, we only have the capacity for 100 people, so some people are joining us from YouTube.
Great.
And she's from Surabaya. It's another city from Indonesia. She has some questions. How to validate drugs dosed from pharmacogenetics, and to know the effectivity from therapy, do we need preclinical study on animals?
Okay, so let's have a look at that first question. I'm going to just look up the pharmacogenetic testing. Let's see if I can find a good example. There are databases now available. Maybe I'll send Angit a link after this meeting, where you can put in a specific drug and it will give you an example of the recommended dose differentiated by genotype. So for a specific person who has an A at a specific location, or a T, or like our height example, if they have an AA, an AT, or a TT, there will be an example of the recommended doses. So for some individuals, like the intermediate metabolizers, they'll need more drug to get that drug working to a suitable level. For instance, a good example is always warfarin, where you take warfarin to thin your blood so that you have less risk of clots. In those individuals, if the drug doesn't work, they'll still be at risk of clots. If the drug works way too well, they'll have life-threatening bleeds and be back into hospital. There'll be some a very continuation of individuals where it works pretty well and it doesn't really work that well, but the side effects aren't as bad. And there is a database, I'll send it to Anki, where you can put in the drug and it'll tell you if this individual has these specific genotypes, they need this recommended dose. If they have these genotypes, they need slightly higher, or they need slightly more, or an alternative therapy. The second question on animal testing with clinical trials is kind of a bit far away from this project, from my work, but I think in general, clinical trials for testing drugs need to be approved in animals before they go to humans, so they're not at risk of killing the humans who volunteer for the trial. But that wouldn't be really in my field.
Thank you. Moving on, so we actually have a lot of questions. So it seems like you gave a very interesting talk.
Oh, I'm glad.
So there's a question from Mr. Sumarmo Marno. So how far has the implementation of pharmacogenetic approaches in drug therapy in the UK, especially in anti-epileptic drugs?
Great question. So it's well known in the UK, but the implementation isn't there yet. So for example, the two examples I gave you about the bedside test and the pharmacogenetics passport are still in very initial stages. So in general, it's not there yet, but it's about, I think at the minute they're all trying to set the scene to allow for this routine genetic testing to allow for more pharmacogenetic analyses. In the case of some of the anti-epileptic drugs, or that example I gave you, HLA variants, it would be quite well known for specific brands that 20% of patients don't respond well to this drug. In the example of warfarin I gave you about blood clots, a lot of the time they'll increase the dose very slowly for warfarin, and there's like an 8-week or a couple of months of analysis of where the patient is put on an increasing dose to check that they handle the symptoms, or they just give them a different brand where a blood thinning drug that it hasn't been shown to have as much pharmacogenetic effect. And that's fine for where there is the option for a different brand. But in general, pharmacogenetics hasn't been massively implemented in the UK yet, but we're getting there.
I imagine Indonesia is still far away from that, but we're also getting there.
I think as genetic analysis gets more routine, we'll all be able to use it a bit more.
Yeah. So there's some more questions. So this is from Institute Technology Bandung. So it's from Bandung. Thank you for the insightful presentation, Dr. Catherine. As you mentioned earlier, most pharmacogenetic study was conducted in children and babies. Is there any specific reason behind this? And if we're going to do one, what is the most important thing to consider in choosing the volunteers? Thank you.
Okay, so I think the reason the therapies in the UK have focused on a lot of pediatric pharmacogenetics is because the disease is so severe and debilitating for those children. And if they can address that when they're young, hopefully that child will have a longer life. For the question on if you want to set up a pharmacogenetics analysis, it depends on what you want to look at. So if you have a specific therapy and you want to see if there are genetic variants involved in the response to the drug or the severity of an adverse reaction to the drug, you need to collect enough patients to have statistical power. So that's really important. So for looking at mutations that are common, that are in 1 in 100 individuals, you don't need as many people to have power where you will find significance in a genetic variant associating with your phenotype. But if you want to look at rare variants, which are more severe, you need to collect a lot more individuals, and that can be very costly. I think for recruitment of pharmacogenetics, of recruiting of patients for any genetic analysis, you want to take as much data as possible. So any variation in the phenotype. Things that usually go under the radar are things like adherence to drug. So if the patient has actually taken the drug at when you said and at the right dose and every day, as you said, as you had prescribed to them. But I think the things you normally need are, for a genetic test, say we do genotyping, which is common variants, you need age, sex, and depending on what phenotype you're looking at, if there's other covariates, you want to include those as well. So for instance, in a lot of cardiovascular measures of the heart, we take into account systolic blood pressure, which can have an effect on the heart that's not due to genetics, it's due to the blood pressure system. Or body surface area can have quite a big effect on MRI images. So we take that into account as well. So normally we would take into account age, sex, BSA, body surface area, systolic blood pressure, and then also ethnicity. So I mentioned that Europeans are very similar, but what you don't want to do is have in your patients versus in your controls, if you have patients that have that are 75% European, and then in your controls you have patients that are 5% European, the test you do might end up picking up just ethnicity specific variants that are specific to Europeans and not actually disease specific variants. Now there are ways of adjusting for ethnicity, but you need the genotyping data for that. So you can't just look at one gene, you'd have to look at the whole genome to try and estimate their genetic ethnicity. And then you can use that to adjust, and you can have any type of ethnicity in your analysis. So I would say covariates, and also depending on what you're looking at, think of ways that the result you get for a specific trait, severity of their adverse drug reaction, how it could be altered by what they've done. So things like if they've taken the drug or not, or at the right timings or elsewhere.
I think for genetic study, you need a very large number of samples.
So, yeah, well, you do, because it's a multiple testing problem. So for instance, for my PhD, I did a GWAS of on lipids, and it was 500,000 individuals, and I analyzed 1,000 samples, and using that 1,000 samples, I got results. But what you have to do is it's like a t-test for every variant versus their lipid levels. So if you run one t-test, you don't need to adjust the p-value, but because I ran 500,000 SNPs, I had to adjust for 500,000 tests, because they're all independent tests. So you have to get an association that reaches a p-value that's very, very, very low. So in my case, it's 8 by 10 to the -5. So it's really, really low, like lots of zeros. And the only way to overcome multiple testing is to have enough patients recruited. So in my case, 1,000 samples was enough to look for common variants of lipid levels, and only really the main enzymes involved in the lipid levels. Now, in things like blood pressure, but also another thing is that lipid levels are a bit more controlled by their metabolic enzymes than the rest of the genome, while blood pressure is continually being analyzed by massive consortiums where they've looked at 1 million people with ranges of blood pressure by genetics, and they're finding the whole genome associates, because there's so many different factors in your body that affect blood pressure. And if you were unlucky and you thought the effect size of these SNPs was quite low, like each SNP in blood pressure has a small effect on blood pressure, you need to analyze lots and lots of people. But if it's something like lipids, where there are specific genes involved, I analyzed 1,000 and found those main genetic variants and those main genes involved in lipid metabolism. So sample size is definitely you need to really think about it. And there are power calculations you can do, but also this is where the whole genome sequencing hasn't been used enough, because if you want 1,000 people, you need a lot of money to do whole genome sequencing, while it's a lot cheaper to do genotyping for more individuals at a lot less of a price.
Very interesting. I would like to ask the committee, do we still have some time or at the end?
Oh, it's already at the end, Dr.
Okay.
Anggit.
Okay, thank you. Thank you so much, Catherine, for the interesting talk.
No
We'd
problem
love
at
to
all.
have you here when the
I
pandemic is
know.
over. You should come here in Lombok. It's a very beautiful place, actually.
I'd love to. And thank you so
Yeah.
much for the invite. It's really brilliant to be able to speak to you all.
Yeah, thank you. I'll give it back to the committee then. Thank you, Catherine.
Okay, thank you very much, Dr. Anggit, and thank you, Dr. Catherine, for sharing your research and expertise in today's events. Thank you so much. Gently remind of all the participants to fill the attendance form that has been put on the chat room because your certificate will be sent based on that. Thank you all. It's already 17, almost 6 in the evening in here in Mataram. Thank you for your participants and behalf on the Department of Pharmacy, Faculty of Medicine, University of Mataram. I will say thank you, and we are looking forward to meeting you for our next international lecture. Thank you. Have a good evening and have a happy weekend.
Do you have a certificate? No.
Thank you all.
Tidak ada
Baik, karena Dr. Catherine McGurknya sudah leave group, jadi kita akhiri dulu untuk kuliah tamu internasional hari ini. Untuk link presensi bisa diisi jika ada yang belum mengisi, karena sertifikat akan diberikan setelah mengisi link tersebut. Baik, mungkin itu saja.
Tidak ada pemberian sertifikat ke Catherine ya tadi.
Sudah pergi duluan tadi.
Iya, sudah lewat tadi orang.
Iya.
Kelupaan.
Oke, terima kasih semuanya.
Terima kasih, Bu.
Ya, sama-sama semuanya. Terima kasih.
Semuanya.
Ya.
Terima kasih, Bu.
Oke.